Thank you very much, Kosuke, for agreeing to give a build cell seminar, and you can get started.
Thank you, Kate, for having me. My name is Kosuke Fujishima. I'm one of the PI at Earth Life Science Institute at Tokyo Institute of Technology. We're actually under a crazy period where the name of our university will be changing in a few months. So actually, from October, our institute will be renamed as Institute of Science Tokyo with a new logo. So please don't be panicked if you see anyone that you know from Tokyo Tech suddenly all changing their name or affiliation to Institute of Science Tokyo. Today, my talk will be centered around expanding the reconstituted cell-free TXTL system, which maybe you're already familiar under the name of Pure System. Just to briefly introduce our institute, LC was started in 2012 as one of the research center designated in the World Premier International Research Center Initiative, supported by the Japanese government. It's located in Tokyo, so if you have any chance of visiting Tokyo, please let me know. I'll be happy to give you a lab tour, introduced to many researchers here at ELSI. So our main theme is actually to try to understand how Earth was formed and how life was emerged on the planet. So we actually have a suite of researchers with a background in Earth and planetary science, geochemistry, biochemistry, and a biologist like me. My background is actually molecular biology and also pursuing synthetic biology. But I also have many interests in the origin of life and astrobiology in general. So, speaking of the reconstituted TXTL system, um, many of you may have already know, but this system is a reconstituted version of a coupling of transcription and translation. The buffer system is actually HEPES KOH, so it's potassium-based with also magnesium added. Interestingly, no chlorine is used because it's known to inhibit the translation machinery. There's also a reducing agent like DTT, but this can also be replaced by other reducing agents, such as glutathione. Also contains NTPs, amino acids, and also other energy regeneration, so energy molecules and energy regeneration enzymes, amino acylation enzymes, um, all the components related to the transcription and translation, such as ribosome and the elongation factor, initiation factor, and so on and so forth. So this system was originally invented by the group from Tokyo University of Tokyo, Ueda Lab, and it's been refined since then. Um, it's a relatively complex system. However, we know all the components in the system, so that's an advantage. If you compare this to for example, a cell lysate system, the yield is relatively low. So usually you will get up to 1 milligram per milliliter of a protein in an ideal condition. So it's relatively low yield. However, it's free of protease and nucleases, so it's actually quite useful when you try to do, for example, like a display type approach. I will explain that in the next slide. So any type of in vitro evolution experiment, this is quite advantageous because of its stability. The other draw, although the other drawback is the cost, it's quite expensive. So that's another issue when handling these systems. Luckily, um, there's a main provider of the Pure system in Japan. And the company name is Gene Frontier, and my lab is collaborating with Gene Frontier so far. So I have the privilege to use some of these components for free. So some of the work that I have been doing in my lab, aside from the expanding the TXTL system, is to actually utilize the pure system for in vitro evolution study. So as I said, I'm my one of my main research interest is to understand the origin of life. So I'm interested in actually creating a polymer, a polypeptide that could have existed in the past. So by using the pure system, we were able to actually refine the traditional mRNA display, which was originally invented by Shostak and Gold back in 1990s. Um, for those who don't know, who are not familiar with the mRNA display, let me just briefly explain. This system, you start with a library of DNA, encoding different, each DNA encoding different polypeptides. Then you transcribe the DNA to messenger RNA, so you create an mRNA library. Then you ligate a DNA pyrimycin tag onto the mRNA, and you subject that to the pure system, the translation machinery. Because the mRNA does not contain any stop codon, ribosome translates the mRNA and stalls at the junction of the mRNA and DNA tag. Then you add salt, which induces the pyrimycin to enter the A side of the ribosome, creating this beautiful mRNA peptide complex. So now you have a phenotype-genotype linked molecule up to 10 to the 12 variety. We refined this method using the pure system, so it's extremely so RNase-free, protease-free makes it extremely stable, allowing us to reach up to 50% of display efficiency, which is 5 to 10 times more efficient than the traditional method. So with this, what we did, if you look on the right side of the slide, we actually challenged to retrograde the ribosomal protein. So this is a universal ribosomal protein 11 C-terminal domain, which constitute of 70 amino acids. This domain is well conserved and well known to bind to ribosomal RNA, helix 3334, with extremely strong binding ability. So the dissociation constant is actually 2.4 nanomolar. Now, what we did was we actually tried to retrograde this into a only using so-called early set of amino acids that are prebiotically abundant. So as you can see here, these early amino acids constitute of 10 different types of amino acids, no basic amino acids included, and there's only 2 charged amino acids, negatively charged amino acids, meaning that we're trying to make this polypeptide, the UL11, into acidic polymer. After 16 rounds of extensive selection, we ended up selecting for a sequence that is fully acidic. However, the dissociation constant remained around the same range, so it's still 8.1 nanomolar. So we were extremely intrigued and surprised by the fact that even without even losing like 11 basic amino acid residues, how come this peptide still binds to the RNA? Turns out, we tried to get a crystal of this complex, but we couldn't manage to get it. It seems to be structurally less stable than the wild type. However, by doing the MD simulation, we found that all the replacement residues of the basic amino acid is mainly converted to acidic amino acids like glutamate. And then magnesium ion basically fills in the cavity. So what that means is the cation is basically bridging the negatively charged RNA and the acidic peptide. So this kind of cation-dependent RNA-protein interaction is newly found through our research, and we think that this could be one of the primordial form of interaction between RNA and protein, which is quite interesting. So with that, I would like to move on to the whole bottom-up self-reproducing artificial cell story. So here, I just wanted to highlight again the components that are used in the TXTL system, and there have been many works done in order to either modify or upgrade these systems by using different components, or for instance, there are a group of work done to swap out the energy regeneration system, or for example, add on a new metabolic pathway in order to support the regeneration of the energy molecules. There have been work done to modify ribosomes or incorporate a modified version of these translation factors. There's also work done by coupling to a DNA replication system or modifying RNA polymerase. There's also work done to modify tRNA or use in vitro transcription tRNA. Also introducing recombinant amino acyl tRNA synthetases coupled to the reconstituted cell-free system. So all of these actually leads to these bottom-up approach to try to make it more sustainable. The largest drawback that I personally think is that currently the whole TXTL system is not capable to reproduce, or in other words, regenerate enough amount of ribosome to sustain the whole translation machinery. So I think that's kind of the bottleneck that we're facing at this point. And the other challenge is actually, sorry, there's also work that's been done showing that you can also couple to phospholipid synthesis. The other challenge, one of the challenges, is to actually couple this translation transcription system to amino acid biosynthesis pathway. So later in my talk, I will explain a little bit about our recent work on the de novo amino acid synthesis coupled to the TXTL system. The other thing that are missing is the metal cofactor synthesis, because this is very redox sensitive. It's really difficult to handle this in a benchtop microtube environment. But we also challenge this issue in our different project. But first of all, let me just briefly touch base on the work that I myself and Professor Wang from Taiwan did 5 years ago in order to use the poly-P, the polyphosphate, as an energy source to regenerate ATP and GTP. So currently in the commercial pure system, it uses 3 different kinases, the creatine kinase, nucleotide diphosphate kinase, and the myokinase known as adenylate kinase, in order to regenerate both ATP and GTP using the phosphate coming off of the creatine phosphate. However, if you think about if there's any enzyme that actually capable to phosphorylate both AMP and GDP, you could actually use a single enzyme. In this case, we actually used polyphosphate kinase from Cytophaga, which was provided by Professor Yakunin. And in order to regenerate these nucleotides to couple to the amino acylation and also translation. The only issue we faced under this system is that the Km, so the affinity of AMP and GDP against PPK2 was almost 15-fold different. So we had to actually balance the concentration of ATP and GTP in the renewed pure system. However, by fine-tuning our system, we're able to showcase that by using the polyphosphate as a phosphate source, we were able to not only produce a slightly higher titer of SFGFP, but also much faster yield of the superfolder GFP compared to the 3 kinase system. under this specific condition. So we can actually try to optimize magnesium concentration, the ATP, GDP, the poly-P concentration. The magnesium and poly-P actually, the balance of these two components are quite important because magnesium actually has affinity to the poly-P. So if you had too much poly-P in the system, it actually starts to eat up the magnesium. And if you eat up if it eats up the magnesium, then it actually hinders the translation because it affects the ribosome itself. So the balance is quite important. Stoichiometry is important. The other thing that we realized is that after around 40 to 50 minutes of translation, the protein yield starts to decrease extremely rapidly. And we tried to understand what's happening in the system. Well, we figured out that the poly-P that we've been using is the 25-mer poly-P. And after, well, it's really hard to tell where it actually start to drop, but at least in between these 90 and 120 minutes, we see a clear drop of the 25-mer long chain fraction completely depleted in our system, suggesting that the affinity of the PPK2 towards the shorter chain poly-P is actually much lower. So the PPK2 actually favors long chain poly-P, and it's actually difficult to process the short chain. We also tested, in order to test this challenge this hypothesis, we tested 100-mer poly-P. Sorry, I haven't shown the data here, but actually the 100-mer poly-P showed 30% better yield compared to the 25-mer, which kind of supports this hypothesis that PPK2 favors the long chain. So this was kind of the story behind what we initially started to work on, gradually expanding the TXTL system. Now we're moving into the cofactor synthesis. So as you all know that the metabolism requires many cofactors. So there are many organic cofactors listed here, but also there are a set of inorganic cofactors. And the most famous one is the iron sulfur cluster, which is often oxygen sensitive. And also these cofactors often requires assembly system. So if in case of iron sulfur cluster, there's a famous assembly system known as ISC or SUF. The iron sulfur cluster protein is ubiquitous and essential protein for life. For instance, in E. coli, there is total 140 iron sulfur cluster proteins found. And based on their functional annotation, you can see that 1/3 is actually used for respiration, which makes sense. Electron shuttle, catalyzing redox reaction. What's interesting is that, for example, if you look at the right side, it's also involved with amino acid biosynthesis, amino acid catabolism, purine and pyrimidine metabolism, and cofactor biosynthesis. So not only these iron sulfur cluster proteins are important for passing around electrons, but also actually are heavily involved in the synthesis of these key metabolic compounds in the cell. And on the other hand, if you're on the left side, it's involved in, for instance, DNA repair, RNA modification. In this case, it's actually used in ribosomal RNA and tRNA modification. So this is also another interesting feature that these modifications, for instance, methylation is quite important for the maturation of ribosomal RNA. And also, for example, thiolation of tRNA is sometimes important for certain organisms. So in order to achieve these reactions in the cell-free system, we have to start handling the iron sulfur cluster. However, the challenges for using, well, there is a challenge for, first of all, using the recombinant iron sulfur cluster protein, simply because the iron sulfur clustering, the major FES proteins such as ferredoxin, has a quite a low KD against many other proteins inside the cell. So when we tried to purify an E. coli ferredoxin, all these different proteins were co-purified together, meaning that it's quite difficult to handle or control the amount and the types of ferredoxin for the cell-free system in a recombinant fashion. So ideally, we want to actually express a specific ferredoxin using the TXTL system. However, the iron sulfur cluster protein is sensitive to oxygen, and also we need to incorporate the iron sulfur cluster assembly system. So when I looked into the previous studies, actually Schwartz group has already challenged this using the cell lysate system. So they were able to culture E. coli in an anaerobic condition, and they were able to actually purify iron sulfur cluster protein in a aerobic chamber. So in this case, the 2FE2S iron sulfur cluster ferredoxin, and they're also able to express and purify iron hydrogenase, which is extremely intriguing. And so although the maturation efficiency was not 100%, this is kind of the schematics that they actually what they did in their study. So in order to assemble, in this case, FeFe hydrogenase, they also introduced overexpressed the assembly system for hydrogenase maturation. So, but these are all done in a cell-free, and it also requires a setup of an anoxic chamber. So this is my postdoc, Shota Nishikawa. Back then, he was a PhD student. He used to work extensively hard with these anoxic glove box chamber. He kind of looks happy, but I think he had a lot of issues it's really for those who actually have the glove box in your lab, you know how hard it is to be there, work all day through with this like thick glove. So the whole point of my talk will kind of centered around how to make Shota happy, how to make him happy by leaving the whole system out of the glove box. And in order to do so, we came up with this idea of the 3-enzyme system, a cascade to not only remove oxygen, but also at the same time regenerate an important cofactors such as NADH and FADH2. So the electron is actually coming off of formate. So we're using the reducing power of formate, which then reduces NADH by FDH, and this NADH is then used to reduce FAD to FADH2, and FADH2 can spontaneously react with the existing oxygen in the solution, generating the hydrogen peroxide. So by adding the third enzyme catalase, you can reduce the hydrogen peroxide to water, and therefore we can remove the oxygen from the system. So this system is able to achieve oxygen removal and do coenzyme synthesis at the same time. So we purified the 3-enzyme. We tested the oxygen level in the pure system, in the solution in this case, by using this oxygen-sensitive dye called resulfin. This resulfin, when it's oxidized, it shows this pink color, but in a reduced form, it doesn't show any color in this dihydroresulfrin form. So by adding the titanium citrate, which reacts with oxygen and removes oxygen, you can see that you don't see any color at all. And when we actually do the 3-enzyme system, when we incorporate the 3-enzyme system, we're actually quite good at removing oxygen from the solution, maintaining the reduced environment. it's quite interesting to see that even without the catalase, this system is maintained as well. Although there should be some hydrogen peroxide left inside the system, which may hinder any enzymatic reaction. So we ended up going with adding the catalase throughout the whole system. So the other side trick is that we try to minimize the headspace in order to minimize the oxygen coming in from the air. This system wouldn't work if you keep the lid open. So you have to make sure that you have to close the lid, minimize the headspace, and just try to remove the entire oxygen in the solution as much as possible. So just for small detail, the headspace contained actually 0.9 micromolar oxygen gas. So we had to actually add 9 millimolar formate in a 0.2 milliliter reaction to eliminate all the oxygen from the solution. But if you can maintain the stoichiometry, technically you can maintain a very reduced anoxic solution. So we checked whether this cascade can work well with the TXTL system. So we mixed these enzymes and formate with the pure system. Surprisingly, the yield of superfolder GFP was not affected at all. So there was only less than 5% difference in the yield of SFGFP. So this was a really good sign, meaning that this system is working with the pure system. So next, in order to reconstitute the iron sulfur cluster, we ended up using the SUF system. We didn't go with the ISC simply because SUF is known to be more slightly oxygen tolerant. Maybe ISC may work with the oxygen scavenging system coupled, but we haven't tested that. So the SUF system consists of 6 different enzymes, 6 different proteins from SUF-A to SUF-S. And this is the optimized condition in order to reconstitute the iron sulfur cluster, as shown in the schematics on the right. So you have the complex of SUF-B, C, and D, which then, and then you also have the SUF-SE complex, which actually obtain the sulfur element off of the cysteine, and also by incorporating the iron, in this case, it incorporates iron 3 and iron 2 because partly iron 3 is reduced by FADH2 to iron 2. So you get a mixture of both ferric and ferrous iron, which then coupled to the sulfur to form a 4Fe4S cluster on the SUF-BCD complex. And this iron 4Fe4S cluster will then be passed on to SUF-A as a carrier, and which then passed on to the recipient protein. So just to make sure that the iron sulfur cluster was formed on the complex, we monitored the UV vis absorption, and we found that this 420 nanomolar absorbance is increasing over time. As you can see on the right, you see a very nice black yellowish color, which is an indication of the formation of the iron sulfur cluster. In order to do a more precise measurement, you have to actually use, for example, Mossbauer or EPR type approach, but we didn't have that instrument in our lab, so we basically confirmed the formation of the iron sulfur cluster based on the absorption and also based on the enzymatic activity that I will explain in a second. So now we have the oxygen scavenging system coupled with the cell-free translation system. Here we focused on the expression of well-known iron sulfur cluster protein aconitase, which is one of the key enzymes in the TCA cycle. So we first translated the apoaconitase under anoxic condition using the O2 scavenging system. Then we supplemented the SUF system with the iron. And we ended up the maturation of iron sulfur aconitase protein was achieved under this two-step one-pot system. So this was published in ACS Synthetic Biology last year. Some of you may have already know about this paper. We ended up providing a cover art of this beautiful illustration of the two-step one-pot system, and which was actually shown as the schematics here. And just to make sure that the enzyme system is working, the aconitase activity was monitored by coupling the downstream isocitrate to another enzyme called IDH, which converts isocitrate to alpha-ketoglutarate, and at the same time reduces NADP to NADPH. So we can monitor the yield of NADPH with a 340 nanometer absorption. So by using a fully reconstituted aconitase, recombinant aconitase from E. coli, which is purified under anoxic condition, we compared its relative activity to that with the cell-free pure system produced aconitase, and we actually achieved up to 70 to 75% activity under this condition. So we're quite happy with that we can actually recreate iron sulfur cluster protein inside a test tube on a benchtop microtube. So just to conclude, this oxygen depleted pure system can serve as an efficient platform for functional expression of oxygen labile metal cofactor protein. So this is not limited to iron sulfur protein. So for example, instance, in the future, we could challenge, or anyone who wants to challenge making, synthesizing inorganic cofactor bearing protein, or for example, radical SAM enzymes, please feel free to reach out to me. I'm happy to provide all the plasmids to express these components to utilize the system. I just named it as pure redox, which I thought it sounds quite nice. And I'm happy if people start using this pure redox to achieve or to kind of expand the whole cell-free system. And this system can also actually, because the O2 scavenging system cascade is coupled to the regeneration of cofactors, if you're also interested in the regeneration of the cofactors, this could also be a nice system to use. And also, yeah, and if you're interested in RNA, like tRNA modification or ribosomal RNA maturation, maybe this system could also help you out. Okay, so how am I doing on time? Do I still have?
You still have time, yes.
Okay, great, thank you. So my remaining talk will be about the coupling of the cell-free system to metabolism. There was recently there was an interesting paper published from Fujiwara Group from Keio University, Japan. They showed that there seemed to be a metabolic tug of war between glycolysis and translation. So they actually found that glycolysis actually eats up too much of the ATP, and therefore the protein synthesis does not proceed. So balancing the metabolism and the TXTL system seems to be quite challenging, even with this kind of a simple addition of a single metabolic pathway. In our case, we try to couple the TXTL system to amino acids biosynthesis, because if we can start making the whole system sustainable in a way that if it can regenerate the amino acid by itself and then couple that to the translation, that could be more I say sustainable. Let me just hide this panel. How can I do so when we consider the amino acid biosynthesis, this is the central metabolic pathway, and glycolysis coupling is already achieved by Fujiwara group recently. But when we think about the amino acid biosynthesis, pyruvate seems to be the key precursor for all the amino acids in downstream. So, and here you can see that pyruvate itself is directly coupled to 6-amino acid synthesis, acetyl-CoA to 3-amino acid, also 5-amino acid downstream of acetoacetate, and the TCA cycle can also produce other amino acids as well. So here, we wanted to think of a way to actually couple the amino acids biosynthesis to the fessile generation of pyruvate. So in order to do so, we focused on using the polylactic acid plastic as a cheap source of lactate. So PLA is a well-known recyclable bioplastic, and PLA's recent paper showed that PLA can be degraded into lactate using an enzyme called PLA hydrolase. So, and also lactate can be converted to pyruvate in a one-step reaction using the lactate dehydrogenase. So by coupling the PLAs and the LDH, we could actually start producing pyruvate in a relatively cheap and accessible fashion. And the other interesting feature about the PLA is that it can form a microdroplet. So my colleague, Tony Jha, has actually worked on forming these microdroplets using PLA. So he made a gel dissolved in 20% acetonitrile, and within this microdroplet, he was able to concentrate several different biomolecules such as thioflavin T, RNA, or even a protein. So we can kind of see the PLA as a source of like a solid or a semi-solid phase-separated feedstock of lactate for the cell-free system. So now converting PLA to the 4-amino acids, we focused on conversion of lactate all the way down to, well, first of all, glutamate coupled to aspartate synthesis and then to aspargine. Glutamate can also be transaminated and converted to glutamine. So we focused on synthesizing these 4 amino acids from lactate. By using the equilibrator, we also calculated the thermodynamic feasibility of the synthesis of asparagine and glutamine, and there seemed to be thermodynamically favorable. In order to achieve this whole system, we actually need to also add alpha-ketoglutarate and NAD, and also ammonia and CO2. So it's quite interesting in a way that this is kind of anabolic and catabolic in some sense, kind of feeding in these inorganic nitrogen and carbon source, but also feeding the organic acid alpha-ketoglutarate and cofactor. And also adding 7 different enzymes in order to couple all these reactions together. If you're interested in, so this was actually recently published in ACS Catalysis. So I wouldn't go into the details of the type of enzymes, but this is the list of the enzymes that we actually used in this research. And what was quite interesting is that even though we actually tried our best to balance the yield, the titer of each amino acids, we were not able to get enough yield of the glutamine. Probably because the Km of the glutamine synthetase, the transaminase reaction seems to be not efficient. We're still working on this. We are thinking of using different enzymes here, but currently this is our best yield. So we have enough glutamate, but very poor glutamine, and also the yield of asparagine and aspartate is not as good as we expected. However, we considered going forward. So after we're able to do the seven enzyme system to generate the 4 amino acids, we then try to couple this to the translation transcription system. So we used a pure system with a customized pure with 4 amino acid eliminated, and to see if the lactate produced using lactate as a starting material, whether our synthesized amino acids can rescue the yield of superfolder GFP synthesis. And as you can see on this graph here, only when these 4 amino acids are added, well, first of all, we check the whether this customized pure system is working or not. Only when the 4 amino acid is added, this customized pure system works. And when we started adding these compounds into the system, we actually start to see the yield of the GFP as shown in this graph. So, but we actually tested different concentration of the alpha-ketoglutarate and ammonium ion. Also, ATP seems to affect the final yield as well. So this is kind of the current status. We were able to generate the amino acid in the cell-free system and couple to the TXTL system. And last but not least, we then coupled the system in order to produce the PLAs by the mRNA translation, and then have the PLAs chew up the PLA gel to produce more lactate so that this lactate can feed back into the system to produce more PLAs. So in a sense, we wanted this whole system to make it in a more self-regenerating fashion of amino acids. So we actually had to add a little bit of starter amino acids, but the starter amino acid was enough to kickstart this whole PLA's production and the production of more amino acids. In order to make sure that this amino acid synthesis is actually working and not detecting the starter amino acids, we actually introduced these isotope labeled ammonium and carbonates. And also we checked the yield of the production of the additional PLAs by doing the Western blot analysis, and we're able to see that after 6 hours, the PLAs is actually generated. And we also did the LC-MS analysis to do the isotope analysis to make sure that the amino acids that we're detecting, it's actually coming from the CO2 and the ammonia that we added into the system. So it seems like we're able to couple the whole reaction into the whole scheme of the translation transcription system. So with that, I would like to end my talk and thank all the members who were involved in the projects that I introduced, presented. So thank you all for listening and happy to take questions.
Thank you. Thank you very much. That was fantastic. I really love this work. There are questions in chat. Can you see chat?
Yes, I can see the chat.
If you could read the question out loud before answering, that's going to help with the recording.
So first question from Christiana. Thank you. Awesome talk. With this redox regeneration system works in TXTL, not pure. Oh, that's a very good question. We haven't tested the TXTL. I think you're mentioning the cell lysate TXTL. My gut feeling is telling me that it should work, although we might have to play around with the concentration or the stoichiometry of each enzymes plus the formate concentration, but I think it should also work as well. Yes. Petra, thank you for the question. This is very inspiring talk. Followed up on the previous question, do you think the amino acid regeneration system would work in whole cell extract too? Or is there something they're limiting it to pure? Let's see. Um, no, I think it should work. So again, I think of course the cell lysate itself should contain a couple of enzymes that are already presented here in this work. So there should be some background. Well, sorry, I'm not familiar with the cell-free lysate. Just if any of you know about, for example, back if there's any background reaction of amino acid synthesis going, I would like to know about. But in any case, um, these are the cocktails of the enzymes in order to produce specific amino acids. So depending on the stoichiometry, I think we should be able to generate these amino acids. But some of these enzymes might already be present in the cell lysate. So sorry, I think there's a lot more to work on, but thank you for the question. Henrik, a question from Henrik. Do you think your amino acid regeneration system could be coupled with other regeneration systems, for example, the energy coupled carbon cycle Tobias really recently posted on preprint? Sorry, I haven't checked Toby's recent preprint. So this is, I guess, different from the Jewel paper, but let's see. Sorry, so I can't answer this question. But Henrik, if you're there and if you can actually speak out, could you actually explain to me about this recent preprint paper?
If you could unmute yourself, or I actually can paste the link for anyone interested. Let me find it. It's gonna take me a moment because as always, I'm bad at finding things. But okay, here's the link for anyone interested. I think, Henrik, correct me if I'm wrong, but I think that's the paper he meant.
Okay, thank
It's
you.
really cool work. It's basically coupling that carbon cycle that Toby's been working on for a while to
Oh, the
energy
CT
regeneration
carbon.
system. Yeah.
Oh, I see. So yeah, so this CTCH cycle is actually the CoA, these acyl CoA coupled cycle, isn't it?
Yeah.
So and yeah, so I think there's an enzyme called CCR that has a really high efficiency in the CO2 fixation. And so I think that in this entire pathway, I wonder where it leads to. So the output of this cycle is going back to one of the
If you pull out
hmm,
figure one, if you open the PDF, the figure one
yeah,
of that
let me
paper
do that.
shows. I'm biased, but I really like that paper.
Hmm.
If he didn't ask about it, I would.
Oh, from the glyoxylate. Oh, this is really nice. Beautiful. I see. So I guess so this carbon, the excessive carbon actually kind of comes off as a glyoxylate, and then I guess um their group coupled that to the glycine synthesis,
Mhm.
and use that to the TXTL system. Wow. I love it. This is
Me
beautiful.
too. It's really
So
clever,
yeah.
and I was wondering if could that feed could I guess okay, I'll Henrik
I could
unmute yourself because I'm basically discussing your question for you. But I wonder if that carbon source could be used in your amino acid synthesis
Ah,
pathways?
let's see. So, lactate. Well, I think so in a sense, our system, so our pathway actually kind of we could couple this cycle to our pathway. So because our pathway is actually producing the glutamate. So the glutamate is actually quite important in order to serve as a nitrogen source, actually providing it's a nitrogen provider for other organic acids or even some other amino acids for transaminase so reaction. So I think we can so yeah so and technically we could couple this system our system to this cycle and then couple to the TXTL system. If there's any inhibition effect, maybe that would be the competition against, for instance, NADPH or NADH. But in this cycle, I think they only use the NADPH. So I think that's fine. Um, they also use, I think, catalase related reactions. So for instance, they actually convert oxygen to hydrogen peroxide. So there's a reaction that uses oxygen and convert to hydrogen peroxide. And this hydrogen peroxide will then be coupled to catalase to get rid of. And so if there's any reaction that does not work well with this cycle is whether if that cycle is more oxygen sensitive, I think. But if we can localize the reaction, so in a sense, if we can compartmentalize this pure redox environment and then couple to CTACH, so you can actually see it as a organelle, like an anoxic or less oxygen sensitive organelle. And so yeah, if we can have like two different environments coexisting, then maybe that'll also work. So this could be still a challenge, but something to think about.
Yeah, I'm really looking forward to that.
Okay, great. Thank you. So, okay, how far did I go? Okay, so Lynn, thank you. Thank you for listening. I recently read you and Kate and I, the review article on the SynBio. I really love that. I really like that table with all the references, by the way.
That table was Lynn's idea.
Oh,
I
okay.
really, I'm really happy how it turned out.
That was very helpful, actually. I read it a week ago. Fantastic.
Yeah.
Daniel, thank you. So, Daniel says there should be no amino acid synthesis in the extract. So this might work. Yeah, I also think so too. Great. Okay, so I think I went through all the questions.
Awesome. I think that's all. I want to say thank you so much again. That was really fantastic, and
My pleasure.
I'm really looking forward for all those systems to finally connect, and we have a self-replicating metabolism.
Yes. Thank you. Okay. One thing I didn't mention about it, but we're also trying to incorporate. So now, since we're handling with the iron sulfur cluster, the sulfur source can also be coupled to the synthesis of sulfur-bearing amino acids. That's what we're thinking right now. So please stay tuned to for the cysteine and methionine synthesis as well in the
It
future.
would be fantastic.
Okay. And thank you
Great.
so much all for listening.
Thank you. Thank you for staying up. I hope you'll get some sleep tonight.
I will. Thank you very much.
Thanks, everyone, and have a great week. Bye.
Bye-bye.